So I think probably most people in the audience are familiar with the NIH, National Institutes for Health. You work with the NIH, or you're not part of the NIH. Talk a little bit about the foundation and how the FNIH fits into this healthcare ecosystem.
Sure. The foundation was actually put together by an act of Congress in the late 90s.
This was back when Congress could act.
I won't say that. But to support the mission of NIH by allowing for more public-private partnerships. And they, primarily what I do is involved in public-private partnerships that are associated with research interests. But we also have partnerships that are involved in training new scientists and new physicians, and doing workshops and things like that NIH would like to do, but just can't get it all together. But we actually are put together in a way that allows us to negotiate science between private entities and the government. And for those of you who have ever tried to do any of that, you realize if you went to NIH and said, I got $100,000 and I really want to give this to you to do some research, and if that researcher at NIH took the money, you would both go to jail, right? I mean, you're not allowed to do that.
And I think maybe back up and explain why. Like, what would be the ethical quandary there?
Well, it puts the NIH and the government under undue influence, right? So, okay, so if I do it, maybe that's not a big deal. But if Merck does it, or Pfizer, or a number of people, that becomes a big deal, a really big deal. And we hear about that all the time in Congress now, right? That there's a perception of undue influence. Well, FNIH is put there as the Switzerland, this space that is neutral, that can be used to gather information from private entities, from the government, as well as any interested party, and help the government either make decisions, but mostly help put together very focused partnerships, very focused research programs to answer and solve very specific problems, right? So, I'm a great believer in if you have a very focused goal, you actually have the opportunity to solve it. And so that's what we do. We put together these. We are not a granting institution that you would normally think of as NIH. NIH kind of gives you grants and they ask you to do your work and comes back 5 years. And we actually are very regimented in our approach, and we expect milestones, and we don't pay you unless you do the milestones and things like that. So we end up with a very clear result.
And it's really about like agreeing on the science. Like you've got the NIH's scientists with their perspective. You've got the private industry, which also has its own scientists, but also has a desired outcome on a lot of occasions. And you're trying to help. Can you explain how you help everyone agree on the science and say, well, look, this is how we're going to test. These are our standards, and these are the markers that we're going to track.
Yeah, this gets to the actual name that we came up for this session, which is Big Tent. I mean, I think people talk. It's team science, but it's team science at a magnitude greater than what you normally think of as team science. So, in the normal situation, you might think 3 or 4 different groups get together from Harvard and MIT, maybe never, but and UMass, and that's a team that gets together. But generally, they all think alike, and they all have the same kind of plan in their direction. What we do is we intentionally bring people together who don't agree, or who do not have the same perspective. So we'll look at industry, government, which have a different perspective. Different places in the government have different perspectives. So we've talked to FDA as well as NIH. They have a very different perspective. Academics, a very different. Patient advocacy, foundations for patients, et cetera. All of these people have a very defined interest in what they want to do. What we do is try to find those areas that overlap all of them, where everyone benefits by working together. And we also do projects that really would unlikely occur or ever be successful if any one entity tried to do it alone, right? So currently, just as an example, it's probably the biggest example that we have right now, so it's not the standard, but we have a program on bespoke gene therapy. It's a bespoke gene therapy consortium, and that was brought to us by the FDA. Peter Marks actually gave us a call and said, this is something I really want to see done in CBER, and we'll be there to help. Companies wanted to understand the path for regulatory decision-making within generating new gene therapy. And obviously patient advocacy groups, patients want to have these things available to them in a way that is actually affordable and doable. And so we spent quite some time, because it wasn't immediately obvious how we were going to do this together. But we now have 15 different companies, I believe, and 12 different institutes of the 27, and a number of patient advocacy and foundations. Many of the foundations, actually, one of the first foundations that started working with us was Duchenne, and they don't consider that rare, really, but they saw what we were doing was going to actually help them as well. So we brought together a lot of people. It did take a bit of time to get it on path, but now we have the opportunity to change the way gene therapy is brought to patients for everyone.
And it's across all the different companies and the government's regulation and sort of approval process. Right. And are there any other examples that you can point to where you're like, this couldn't have been done without FNIH's involvement?
So there's a lot. I mean, I think we have several programs that were brought to us, that we helped develop, that have had really significant impact in the space of healthcare, delivery and development of therapeutics. So the three that I can think of that are kind of hallmarks are Biomarkers Consortium, which is a group of folks who want to run trials for their therapeutics, but can't because there's no good way to measure some of these things. And we've been talking about this today, and so there's a lot of interest in our Biomarkers Consortium to look at digital technologies and the kinds of things that we were talking about. So essentially, everyone here is a potential, I think, a potential person to talk to and be collaborative with within the FNIH.
Maybe just define biomarkers for in their role in the
Yeah, so biomarker is any molecular response that reflects a physiologic entity in biology, and it could be just standard heart rates. That's a biomarker. It's an old one, and it works for everybody. And then there's those that are specific for people who have a disease. So we heard about A1C, right?
Mm-hmm.
That's a biomarker. It's a very good biomarker. It's a very standard biomarker. We don't need too many new ways to do that, but it's you could imagine there are a number of areas where that is useful. So in that case, probably the one that's the most stunning is we have a NASH program. So NASH is non-alcoholic steatohepatitis. It's a liver disease, and if you get NASH, your liver's fatty and fibrotic. But if it gets worse, you get cirrhosis, and there's only really one way to fix that. You get a new liver, all right? So it's pretty intense. And so if there are ways to address that ahead of time, well, right now, the only way that you can diagnose whether you have NASH is to get a liver biopsy. And I don't know if anyone in this room has ever had a liver biopsy, but it's a 6-hour ordeal. There's mortality. You can die, actually, from a liver biopsy. And it's not a large number, but there's a possibility, and you can't move for 6 hours. So you're laying on a gurney for 6 hours because if you start moving too fast, you could bleed. And that's a horrible thing. So what we've done is put together another huge group of people, both academics, industry people as well, and government, FDA is there. And now we have a set of approaches that include both imaging and uh, molecular, blood tests that we heard about that can reduce the number of people who need to get um, biopsies by half, right? So if you're trying to do a trial, that's a huge amount of money. It's a huge drain. It reduces a huge drain on the patients because it's not fun.
So what happens, I see you playing this role as like traffic cop with private industry and the government and trying to align interests and align perspectives. What happens when there's like a breakdown and it's impossible for you to sort of make everybody play nice together, or even the private sector players who all have their own interests?
Yeah, it's a relationship management web of intrigue in some ways. We have had projects where, well, I should say, bottom line, there are some fields that just are not ready to collaborate in the level in the state
Like what?
that we do. I'd rather not say, but there
are cameras rolling. I had to try.
There are some groups of people who do not want to, and we heard about it today. Everybody wants their own data, right? Their own little data heap, and there are some groups that is their livelihood, and that's they don't want to share. And
I think you're actually showing what makes a good traffic cop is someone who doesn't take the bait when you ask that question on stage, because maybe you've pointed out examples where things fall apart, and then a couple of years later, the situation changes, the industry changes, and then people are ready to come together and work on a problem.
Yeah. And then and there have been projects that we started out with people not wanting to be in the same room, actually to talk.
Like who?
So that was a diabetes project many years ago, right? But even the NASH project, it was imaging and blood, right? And they were like, we know what we're doing in imaging and we know what we're doing in blood. We don't need to necessarily talk so much. But obviously they were gonna. But it turned out after several months of they came to us and said, we really need to work on this together. So that was the ideal, right? I mean, we just helped them direct towards the recognition of this is something we need to do together.
I think, and I know you didn't work directly on the project of developing the vaccines, but you have a lot of insight into how the COVID vaccines were released and the timetables. What are your key learnings for that process? Do you think it went as well as it could have, and what made it possible to develop them and release them on such an accelerated timetable?
Yeah, so can I step back a little bit from that? So as we all know, in 2020, there was an event that certainly impacted us all. And being part of the Foundation for the National Institutes of Health, doing lots of large collaborations, my supervisor at the time, my boss, got a phone call from the director of NIH on a Sunday afternoon, and said, there are too many trials. This is directors, Dr. Collins. There are too many trials, and none of them are going to actually finish. We need to actually coordinate. By the end of the week, we had already had 30 or 40 people in a room, many of them R&D heads of the CDC, head of NIAID, so Dr. Fauci was there, and a very large contingent to start to try to coordinate the process. During that process, it was decided there were several areas that really needed to be focused on. One of them was vaccines, and I'll get back to that, but that was actually being handled very much by what ended up being Operation Warp Speed, right? We were kind of advisory kind of area with that. We didn't have that much to do with that, active didn't. There was a clinical therapeutics group that tried to find drugs and test things like ivermectin and others, as well as a preclinical group, because we were finding out that there was a huge need for BSL-3 labs. So these are the labs that can contain these pathogens, and many companies, many people were getting into this, and they didn't have it, and they didn't know where to go, and so we needed to help that. The real one that surprises me was there was a we needed to have a group that just identified clinical trial capacity.
Mm.
Now, you'd think the NIH would have an idea of every single trial site that they have, and each institute does, but they don't have it across all of the institutes. And so it was quite an endeavor to pull together what was out there in terms of being a clinical trial that could affect clinical trial sites that could actually have an impact on COVID. And then we had a group that actually set up master protocols and ran clinical trials for therapeutics. And we ended up actually testing 37 therapeutics. So that's where that goes. So understanding we were close to the action of the vaccines, but we're not there. What really made the difference for the vaccines coming out as quickly as they did was years of prior work, number one, right? So none of those things were like brand new technology. They were brand new technology coming out to people, but there had been a lot of work on them. The other thing that the government did, and this would not have happened without Operation Warp Speed, was parallel processing, essentially. Normally so I didn't say this, but I've been at FNIH for almost 8 years now. Prior to FNIH, I was at Merck for 11 years, and I was at Pfizer for 11 years. So I'm a drug guy, right? I mean, that's my background. I'm an industry drug guy. Normally, when you are running trials and you're getting ready to put stuff into patients, a therapy, you do things sequentially. You do animal testing, and then you do toxicity testing, and then you do what you need to in order to do formulation, and you test that. And you do all of this sequentially, which takes years, right? What the government allowed, the vaccine and the manufacturers to do is to take all of that and do it at the same time. So normally you wouldn't do that, because if the animal testing looked bad, you wouldn't go on to the next thing and you'd save money. But they knew we needed it fast, and so they did this parallel processing and took on the risk. And in that case, in many cases, it was, well, you can see what it did. I mean, I think it was a huge success in that instance, in terms of getting those things out. But in reality, none of the testing was compromised. I mean, we didn't do anything faster than we normally would have done. We just did it in parallel.
Can we do that for other treatments going forward? I'm sure the industry is like, why can't we do this every time?
Because it costs a lot of money.
Because
there's that. Because if one of the first steps fails and you're already starting some of the later steps that cost, generally, I mean, the other thing is generally drug development, the earlier it is, the cheaper it is. And as you get closer and closer to going into humans, it gets increasingly more expensive. And so the stuff that if you're working on those relatively inexpensive things and they fail, you don't want to do the most expensive things, right? I mean, that's just makes sense.